Welcome to OmicsBox, Bioinformatics Made Easy.
Welcome. And this is Stefan Goetz from BioBam speaking, and our webinar title for today is Comprehensive Biological Pathway Analysis, which is about integrating and analyzing pathway and expression data in OmicsBox with a couple of practical use cases. And this will take us about 45 minutes, and then we'll have time for Q&As. I already mentioned Q&As can be written in our Q&A section. And yeah, we will start off with a presentation. I will be doing that, and then we will hop into a practical demo because today's objective is really for you to see OmicsBox in action, OmicsBox working, how you can use it for your data analysis, all the features and functions, all those little things you can use in OmicsBox, which are sometimes difficult to find. I want to show them today, okay? So, and with that said, I will start sharing my screen over here, and I will begin the official webinar now. Let me move this away over here. That's it. Okay. I hope everybody can see and can hear and see well. And let's start. Okay. So first, I will I will present myself. So my name is Dr. Stefan Goetz. I'm working here at BioBam. I have a PhD in bioinformatics and a computational background. So I'm having some 15 plus years in functional genomics research. I'm actually the author of the BLAST2Go methodology and also founder and CEO of BioBam. So today's content will be about different, it would be transcriptomics and functional data analysis. So it would be out expression analysis and functional annotation of non-model organisms data. And then this is kind of the basis for what the actual title is, which is comprehensive pathway analysis and pathway enrichment analysis. So we will cover those two topics as well. And to make it not really too dry, I brought a couple of use cases, two examples. So I have a bulk RNA-Seq dataset of lactated mice, which is actually a model organism, and it's a classical RNA-Seq bulk RNA analysis. And then I have a de novo transcriptomic study, which is about a roundworm affecting chicken. So that's our nematodes studies in chicken. So by using those, we will not really go too much into the biology, but by using those two, we will explain all those features and functions in OmicsBox, which are useful to analyze those types or other types of data sets. Okay. And I want to keep it practical today. So I have some slides, and then I really want to open up OmicsBox and show you around all those features and functions, which hopefully might be useful for some of your studies. Okay. So a quick introduction into BioBam. So BioBam is based in South Europe. We are a bioinformatics software company, let's say. So we have this interdisciplinary team of bioinformaticians. Some are more on the biological, biotechnological side, who are actually relevant for curating and maintaining and adding all those features, functions, algorithms, and pipelines which are available in our platform. And on the other side, we have those computational experts who are taking care of visualizations, high-performing cloud high-performance cloud computing, front-end, back-end issues, those kind of things that makes everything work smooth. Our main mission in BioBam is to close this technological gap, which we believe lies in between the computational biology research and the molecular research scientists who actually want to use those tools which are getting published basically every day. So we have this motivation to make bioinformatics a little bit more easy by closing this gap so that people with less bioinformatics expertise are also easily capable of running those complex bioinformatics pipelines, okay? And we are doing this, as I mentioned already, over 10 years now on an international scale. So we have clients all around the world using OmicsBox, wherever some genomics, transcriptomics, metagenomics research is being done. So that's basically universities, research institutions, biotech and pharma companies. Okay, so our main product is actually called OmicsBox. I guess quite a few of you are already familiar with BioBam and OmicsBox, so I'll keep this short. OmicsBox is an end-to-end NGS bioinformatics platform. So you can put all this raw data formats you're getting from the different sequencing technologies, short reads, long reads, single cells, those kind of stuff, into the platform, and then you can pick the modules we have in OmicsBox and run through those different scenarios to get to the end, which is your biological interpretation, biological insights, to make sense out of your research, right? So OmicsBox is a user-friendly operation system independent desktop application. So it's actually installed in your computer, but then operates on a cloud backend. So all the big number crunching, computationally heavy algorithms are actually offloaded into the cloud, so you don't get issues on your local system. So this is quite adapted by the scientific community with over 15K scientific research citations, a lot of years being out there, and basically used for research, for agrogenomics applications, biotechnology, ecology, and also biomedical applications, biomedical research. We are kind of proud and popular that we are species agnostic. And that means that with OmicsBox, you can run any kind of species through your pipelines. And this means a lot of non-model organism research is being performed with OmicsBox because you don't need those previous functional knowledge to work with the platform, and you can, whenever you're coming with something strange or something new or something off topic, you can basically load in all the resources you need within OmicsBox, and then can do all the complex data analysis as well with the platform. So versatile, user-friendly, and ready to use to go end to end. Okay, so the different modules we have available in OmicsBox are the genome analysis module, everything about DNA-seq, basically. We have our genetic variation analysis module, which is for variant calling, phasing, filtering of SNPs, filtering of variants. We have genome-wide association studies called GWAS. We support population structure analysis, those kind of things, right? So this could be DNA or RNA, and we have different algorithms for different scenarios, polyploid, diploid, those kind of things. Transcriptomics is all about bulk RNA and single cell and long read transcriptomics. We will see in detail today. We have our functional analysis module, which is the oldest module in OmicsBox, where you can do all the basic sequence analysis things like blasting sequences, for example, high performance, high throughput, obviously, but also the more visual and more complex things like the combined pathway analysis we will look into today as well in detail. And then off topic for today, but quite popular in OmicsBox, is the metagenomics module, which is about mixed samples and then doing taxonomic classification and differential abundance studies, those kind of things. So just for you to get a better idea, all those different pipelines, or let's not call them pipelines, like this schemes, for those modules can be represented in this visual form, where you have the little dark boxes as the algorithms available and maintained by us in the platform, grouped by the different topics. So here you can see alignments, assemblies, post-processing, pre-processing. And then those, you can get into those by using raw reads. And then it's not a fixed pipeline, like if you would maybe if you might be using when running Nextflow or some pipelines, but it's actually like just tools available for you. And there's always like certain input requirements, and then you get like the raw output as well. And then you can put one into the other, and then you can like interactively run through your data analysis, going from one step to the other with OmicsBox. That's kind of the big difference here, that it's an interactive platform where you can use your normal standard bioinformatics data formats, and then you pick an algorithm, you run it, you visualize it, and you have all the options in OmicsBox to like fine tune your results, see if everything went well, and then you take the output and you go into the next step. So you have full transparency of what you're doing in each step until you really get to this biological insight, and then you can loop back around to certain, like rerun certain steps if you're not convinced about the parameters or about the output or about whatever you saw on all those many visualizations we have. So as I said, this kind of schemes we have for every module. And as you can see, like for example, genetic variation 1, 2, 3, 4, 5, 6, 7, 8, 9, and more. So there's quite a lot of different algorithms you can stitch together for your analysis processes. For example, here you can use the genome analysis module, do the alignments, then you get your BAM files, and then you go into FreeBias or BCF tools, do the variant coding, and so on and so forth, until you combine your GWAS together with Magma for the functional enrichment, and then you can kind of link genotypes and phenotypes here for your genetic variation analysis and identify those areas where your SNPs are functional or affect your phenotypes. Okay, and then you have the variant annotation and the popular structure analysis as well, which can be used out of the box here. The transcriptomics bulk RNA-seq analysis is pretty well established in OmicsBox, exists for many, many years, always gets updated, of course, but it's kind of a classical pipeline because it's industry standard, so everybody does this kind of things. So you go raw reads, pre-processing, assembly alignment, quantification, difference expression, and the functional analysis. So that's what we're going to be seeing today with the focus on the pathway. So I'll not spend much more time on this. For the transcriptomics single cell part, maybe a few words because that's a pretty cool set of tools we have in OmicsBox. So you can take 3' or full length, so that's UMI based technologies like 10x genomics. You can put it into like the raw formats. You can put it into OmicsBox again, do the quantification, and then from there you can, and you know that there's quite some trouble with single cell because there's a lot of different formats and standards and types. So OmicsBox supports most of them and can handle them pretty well. So if this first step is, if you manage to get that done, so then you can start doing the downstream stuff, which is like clustering, cell type identification, trajectory inference analysis. And normally this is an interactive process where you fine tune, you recluster, you subcluster, you subset your data sets, you do interactive visualizations using UMAPs or t-SNEs, different flavors here for the different visualizations. And then you go, and once you have your cell types, there are also different algorithms available for the cell type identification, reference-based, non-reference-based. Remember, we are always supporting our non-model organisms community here in all steps. And then we have our difference expression analysis, identifying which genes are up and down regulated among certain cell types. And then you can go again into the functional analysis, which we are looking through today as well, which can also be useful then for those kind of pipelines. Okay. So, and then we will, a quick introduction to the long reads transcriptomics module we will not be seeing here today. We had a couple of webinars already, which you can look up in our YouTube channel. There will be also coming new ones because there are new things to be announced here. But basically, we support nanopore and BacBio. And then depending on the technology, you combine Minimap with Flare, and then you can go ISON pipeline for, again, like the non-model community where you have no reference genome. So it's a reference-free isoform reconstruction pipeline available here. If it's a PacBio, so you go IsoSeq, and then you go into Scanty, which is kind of the standard nowadays for transcriptome curation. So there's different steps like filtering steps and a lot of visualization features. So Scanty allows to get this high quality, highly curated isoform resolved transcriptome, and with that you can go into the difference expression analysis module for different transcript usage or different expression, different transcript expressions, those kind of things which can be done in OmicsBox as well. So kind of again here end to end for long reads in OmicsBox. I think we are one of those few platforms who support those kind of pipelines already for long reads. Commercially well created, well established. So if that's something you're looking into in the future, this might be something worthwhile looking or exploring as well. And then the functional annotation and analysis module. Let me spend a couple of minutes here because that's what we'll be using a lot today. So the functional annotation and analysis is two big blocks. So we have our first block, which is combined functional annotation. So this is all about finding and assigning functions or gene ontology terms or enzyme codes or pathways to your proteins, transcripts, or genes or CDS data. So you basically put in the data, which could be output of any of the other steps I already showed, or something you just got from somewhere, from the internet or from your projects. And then you basically are able to run it through a whole battery of algorithms. And there we have like standard ones like Diamond Blast or NCBI Blast, or the InterPro scan for protein families and domains, or we have EggNog for orthology and phyology, which got pretty popular for high throughput functional annotation. We have the RFAM to identify non-coding RNA if you made your transcriptome from scratch. Or you have the subcellular localization support with B gives you some additional GO terms for the cellular component branch here. So everything, each of those steps has a lot of different visualization options, and everything can be merged into one annotation set. So this is something which is super useful if you have a novel or newly characterized genome or transcriptome, because you can manually curate and you can automatically annotate and manually curate your annotations with a high level of transparency. So it's not like AI style black box. You put something in, you get something out, which you never will be able to understand. But here you can really see what's going on, and you can look through the intermediate result and then be confident about your final annotations depending on your downstream data analysis objectives. So for the functional analysis, which the functional annotation is kind of necessary always, we have a lot of options. So once this is done, we can go enrichment analysis. You know that very well. We have the combined pathway analysis. You know databases like KEGG, Reactome, Gramini for plants. The combined pathway analysis I will explain today, and it's about linking those things together. And then we have our quantitative analysis, just looking at counts, looking at geoslims, looking at geographs, see where my annotations are, how my annotations are distributed, bar charts, pie charts, graphs, those kind of things for subsets of sequences, enriched sequences, overrepresented genes, those kind of stuff. And all the data manipulation features like renaming, ID mapping, filtering, selections, and so on and so forth. Okay. So, and last but not least, little off topic today, the metagenomics module for those who are doing microbiome data analysis. This is super easy in OmicsBox. You take your 16S, 18S, whole shotgun or ITS datasets. You do high throughput quality control and assessment of your huge datasets. And then you put it into Kraken. You pick one of those many databases we support. Just for those who are interested in this, let you know we have Midas, Unite, Silva, Green Gene, RefSec. So there's a lot of different databases. And for those who already worked on those kind of analysis, the database selection is one of the most important things. You want to make sure what you're looking for is also in your database. Databases can get really big, so you need efficient algorithms with a high resolution. So you want species level or subspecies level results here. So with this kind of scenario, that's possible. Depends also always on your data quality. But basically what it gives you is a list of taxonomies with quantifications for your samples, and then you can do statistical tests on them, like differential abundance and all that. So you will get everything, Shannon, Simpson, refraction, all those different plots to check for quality, plus your taxa results as well. So I will not spend much more time here, just for you to know that's available and that's super easy to use. So let us talk a little bit about transcriptomics now, because transcriptomics is what we'll be covering here today. So you know all super very well what this is. So it's just a reminder. So normally you have RNA-Seq data, you find expression profiles, which gives you gene lists. You want to go into the functional interpretation, and that gives you the biological insights. So even though we have now spatial single cell and long reads and also things, I think the transcriptomics is still the core technology for like bulk RNA, especially the core technology for functional genomics. So it remains definitely the dominant use case here. And as far as what I can observe from all of our clients, like the functional interpretation remains often still the key bottleneck, the most tricky part here. And that's where we want to add value to your research. So you will be able to do this more easier, more better, even for those species where no functional information is available. And and the whole journey from raw data to biological insights is still not trivial. It's basically not. I mean, there's so many things can happen on the way which can mix up your functional interpretation. So that's definitely something complex to do, and OmicsBox helps throughout the process to get things right. So those two use cases I would like these two use cases we want to see today throughout the demo are basically two. One is a drug response transcriptomics. So here we have a nematode which is affecting chicken. It's called Ascaridia gali, and it developed some drug resistance, which is kind of raising concerns among chicken farm owners. It's a poultry parasite, and has completely unknown genome, unknown transcriptome. So what they did, they just sequenced the nematode from chicken being treated with a drug. The drug is called flubendazole, and chicken which are untreated. And then they were doing Illumina high-seq 2500 short read paired and setup, pretty basic. And I picked this because it's a small data set and it's easy to show, but it's still nice to understand. So I think that's something which some of you might be able to relate to. It's small and has a easy experimental design, but that doesn't mean that in OmicsBox you can't scale up your analysis to hundreds of samples and experimental designs with a lot of different columns and conditions and those kind of stuff. So this will be our non-model organism example here for de novo RNA-Seq. So here we have our experimental design. So we have 6 samples. It's 12 FASTQ files in total. I don't know, 100 million of reads, 2 conditions only. And the typical pipeline for the de novo transcriptome assembly, annotation, and analysis approach is to do obviously the quality control pre-processing. We'll be doing assembly and post-processing, not today, but I'll quickly show. So this is clustering and off protection included. So then we do the basic difference expression of transcripts. So that's just HR or DSEQ functional annotation because it's a novel transcriptome. So you don't know anything about those transcripts you just identified fresh. And then we will do the combined pathway analysis and the enrichment analysis to see what's actually going on here biologically. Okay, so that's dataset number 1. Dataset number 2 is use case for a developmental transcriptomic study. It's actually about mice lactations. So we have this epidermal growth factor signal, which helps keeping milk-producing cells alive during lactation by increasing this protective protein called MCL1. So this is this Nature Cell Biology study, which we took to show OmicsBox, and they were taking different tissues from the mammal gland, and then they were looking how and different mice, like pregnant, virgin, and lactating mice, and then they were seeing how the transcriptomics profiles are changing in those tissues and over the time here to identify what triggers the process of maintaining the milk-producing cells over time, so lactation can continue for feeding, right? So the experimental design here is as followed. We have 12 samples here, those are double. And we have different stages, virgin, pregnant, lactate, as I said, and 2 different types of tissues. So we have now two columns for the metadata, let's say for this mouse. So we have a mouse dataset, which is a model organism. So we don't need to worry about the functional annotation, at least not that much. So this is something we can get online. So we can import gene ontology terms and enzyme codes and so things. So we don't have to worry about that, but still we have to do the difference expression analysis, and then we can directly hop into the combined pathway analysis to identify and see what enzymes are triggered and which genes are up and down regulated and so on and so forth. Okay, so we will be doing those kind of steps: alignment, quantification, difference expression, and then pathway and gene ontology enrichment analysis to identify affected biological processes. Okay. So the use bioinformatics tools, I already mentioned. This time it will be the transcriptomics workflow and the functional analysis module. So if you would want to subscribe to OmicsBox at some point for this type of analysis, the modules needed would be transcriptomics and functional analysis module, okay? So since in the demo, I will be not going over the basic RNA-Seq data analysis steps. So I just put up some slides here for you to see. So the pre-processing is basically you go into the general tools in OmicsBox and you select your FASTQ files. You select whether if it's single end or paired end, and then you get your FASTQ-C report batched for all of your samples. You have the whole list, and then you get your report, things which passed, things which failed. You can plot for all the different characteristics charts, and then you can see what happened here. So here we see perfect read quality. This had already been filtered, already been taken care of, adapters and those kind of things. So there's not really a lot to do. So for the alignments, here we have a star at the gap mapper and BWA as an ungapped mapper. So we can use either of those in OmicsBox. So remember, in OmicsBox, you pick an algorithm, you provide your input data, things get sent into the cloud, things get analyzed there. Once your results are ready, it gets back downloaded into your client and then removed from the cloud if you wish so. If not, you can keep a copy of them as well in the cloud for further analysis steps. Okay. So once the alignments are done, you can do a lot of different quality checks here as well. So you have the gene body coverage plots, you have junction annotations, for example. So there's like Busco and Quast here available for BAM file quality control. And for a de novo assembly study, we would not use an aligner, but an assembly tool. So we would use Trinity, for example. So you can set up those pipelines in OmicsBox, but you can, and that's what we normally recommend when doing something from scratch or new, is to go step by step. So I put here again the Trimomatic and the FastQC step, which sometimes you have to go around a couple of times until everything looks good. It's always good whenever, like most of the times from your sequencing facility, you get your samples already quality checked. It's always still a good idea to recheck, right? I mean, even you get those fancy reports and you have all the plots there and everything looks great. Do your own check because it's not always what you get is what you see is what you get, right? So your own FastQC check is important. And then you have tools like Trimomatic or Cutadapt in OmicsBox, super easy, really fast because it's all parallelized. And then you can do these pre-processing steps. So then you do de novo assembly. So that's basically the Trinity, butterfly, inchworm, and this kind of algorithms which you can run on your data. You get your transcripts. Remember, you're always getting a lot. So you have to do clustering, you have to predict coding regions, and then you have to go into your classification of transcripts, basically by doing blasts and filtering and annotation and those kind of things. We will see in a second. So in the transcriptomics module, you can see the assembly option here where you have all those different things. You have Busco, you have TD-Head, you have the protein predict coding regions, those kind of tools, all readily available for you to run and all the other things. Okay, so once we have all our genes or our transcripts, we have to do the quantification step, okay? So you know that also, but I just want to show you how that's done in OmicsBox, because that's like the thing you have to do before you can go into the difference expression analysis, and it gives you the count table. And if you have all this covered already, that happens to quite a few people. They, oh no, all this is already set up with our own pipelines or our other tools, or we get it from our sequencing facility. So that's fine. You can just take your count table and go into OmicsBox and then start your functional analysis from there. Okay. We do recommend to repeat, I'll explain in a second, but we do recommend to repeat the differential expression of the time course analysis because we need raw counts for a lot of different visualizations later on. I will remind you about this in a second. So the quantification can be done, our use case one, it's, remember the nematodes, so it's a transcript, it's a de novo transcriptome. So we don't have a GFF or a reference genome. So we will just take our list of transcripts and our FASTQ files and we map back to that. So we get our transcript level quantification here. And then we can see how well things align and our library size per sample distribution. So there's a lot of quality checks here as well. So our second use case, the model organism mice study, use case 2, there we do have the human reference genome GFF file. So we just do our alignment, our BAM file with star. That's pretty easy, pretty quick, or BWA. And then we'll get we align things to our reference genome. So we have our FASTA reference genome, our GFF, and then we get things aligned here, and then we get our quantification. So this is I skipped a slide. So the counts are actually shown here. So here you have all your features. So this could be genes or proteins or also transcripts. So here we have our Trinity transcripts, right, as they came out of the assembler. And then we have our counts. So each sample and its counts, okay? And this gets converted with a differential gene expression data analysis into a table where we have, again, the same IDs, the same sequence names, but then with the statistical values, which for HR, for example, is the FDR, fold change, log fold change, and so on. And then we can see also different plots, like heat maps or volcano or MA plots, to then identify those genes which are up and down. This one is also fully filterable and stuff like that. So you get your tags, as in a lot of tables in OmicsBox, we love tags. So you get tags here, so you can filter tags, you can extract tags, you can search for tags. So there's a lot of options. And then we get into our pathway, and that's just a teaser because we will see that in action in a minute. So once you have these difference expressed genes, you can go into this. But you cannot get this stuff if you do not have any functional annotation. So functional annotation is a must to get this, okay? And how do we get this functional annotation? By generating it. And there we come back to our functional annotation and analysis module in OmicsBox, which I already explained. So in OmicsBox, you have a lot of different algorithms, but also databases like gene ontology or InterPro, EggNog databases, which we can use to generate high quality functional annotation labels, okay? And OmicsBox allows you to combine all this information. So how it is done, this is a slide which is really, really old. You are very well with those concepts. So you have uncharacterized genes. You do all the different algorithms, apply them on your data, and then you link it to functional information, which is normally gene ontology terms, and then you transfer them back to those genes, which is the annotation process itself, and that's what got published and which is very popular, which is the BLASTO annotation methodology, which is available in OmicsBox as well. So then you can annotate your genes, and then you can use those. OmicsBox is kind of an interactive toolbox for adaptable functional annotation strategies. So here, each of the individual steps add information to a project, then sequences change color, and you get your functional labels, and then you annotate, and then you add InterPro and EggNog, and then at the end you have your fully functionally annotated project, which you then can work and use in your downstream analysis, like enrichments or pathway analysis. Okay, and this is something I will show also in a second. So for the functional annotation workflow, um, this is like this kind of scheme, which is like the default one, which we recommend, which is basically you take your FASTA files, you run all the different algorithms, and then you do the mapping, and then you merge things back into one project until you have your validated gene ontology annotations here. And there are multiple, you can do this, but you could also go just for EggNog, and this gives you valid annotations as well. A lot of people just use InterPro because they just like the protein family approach for functional annotation, or uh, some do the most conservative or baseline annotation, which is just blasting, taking the top blast hit, and then mapping that back onto your sequences and do that. So those multiple strategies, they can be used like InterPro only, EggNog only. You have the iterative approach where you go from permissive to more strict, for general to more specific, and anything in between. So there's a lot of different flavors, and it really kind of depends on what you want to achieve in terms of coverage or sensitivity or precision as well, maybe. So here's a lot of things you can explore. That's how OmicsBox looks like, and we will see in a second. So you always have, just for those who have never seen OmicsBox before, a quick introduction. So you always have your modules over here, which with all the different options. Then you have your table. Each analysis step, like count table, pairwise difference expression, enrichment analysis, all pathway analysis, all those give different tables with different tabs and different columns. What's the same is you can always save data, export data, and you have your side panel for downstream data analysis options. So you have always the export option, you have always a summary report option, and you have always a certain set of tools you can run on this table, okay? Still, all this can be used with standard bioinformatics formats, and it's all saved on your disk or in your cloud storage. So this can be easily passed around and shared. Those kind of things are available, obviously, as well. So for each of the annotation steps, you get individual visualizations like gene ontology terms with graphs, blast results, InterPro scan results, the gene ontology mapping where we have your accessions linked to gene ontology terms with their gene ontology evidence codes. So kind of stuff very well curated. All the tables have different context menus. So in the context menu, you can show intermediate results, you can change annotations, you can find pathways, you can look at the gene ontology mapping graph to see how annotations have been produced. And then common for all the tables is the copy cell or copy row, and then the creation of ID lists. So you can select a cell, a subset, and then you can just right click and say, create a list of those kind of sequences, or plot any kind of distribution charts for any kind of number values you have in those columns, okay? So the side panel, here you can see the whole annotation process you saw in this picture. Blast, InterPro, EggNog is basically available from the side panel. So InterPro, mapping, Blast. And when you open it up, you can see like chart visualizations. You can remove results again. You can go back and forth and do those things. The functional analysis as an introduction. So we have two options. We have the Fisher's exact test, which is comparing list of genes, remember, and the GSEA, which is using a ranked list, not comparing lists of genes, but having a ranked list plus the annotations, obviously. And then you can get this kind of distributions where you have annotations who are skewed to one side of your ranked list. So this is an overrepresentation of this function among upregulated genes in this case. So for the functional interpretation, there's a lot of different visualization options. So you get the classical bar chart, you get word clouds, you get the popular bubble plot, it's called, where you can see like statistical statistical significance plus the group size, like how many sequences are annotated to certain functions. And you have your charts where you can see how certain enriched functions are and statistically significant functions relate to each other, because often they have a parent-child relationship. So if you have a long list of functions, you can prune those, remove those, so you stick to only the most specific enriched functions, which helps with the interpretation step as well a lot. And now the combined pathway analysis is which we want to see in the demo more. So here you have sequences, annotations, and differential expression. And this means you have this as input and expression. And this allows you to identify, like link your sequences to your pathways, then perform enrichment by taking into account the differential expression, and then generating those maps highlighting your expression data. Okay, so and this is super helpful if you're starting from scratch with a data set, or if you have no clue what's going on, or even if you have a hint and want to search for specific pathways and see which enzymes are linked to which transcripts or which genes, and then look at the experimental design, the raw counts, and how this actually maps onto, for example, here the galactose metabolism pathways, which was relevant for the nematode drug study, flubendazole drug study. Okay, we will see that also in a second. So maybe explain a little bit more in detail the table. So each row is a pathway, and then it indicates which type of pathway database it is. So there are different types. So we have here, for example, KEC, but it could be also Reactome or Gramini. And then we can see how many sequences are annotated to this pathway via their enzyme codes or gene ontology terms. And then I can see how many are actually differentially expressed. So here I can see 4 differentially expressed sequences out of my 61. And they are actually among my uh, list of down-regulated genes, because this pathway turned out to be down-regulated. So I have my underexpressed genes in my drug condition, which caused the, maybe I can show you in this slide here. Sorry for hopping around. So here I had down-regulated genes and had a certain function or a certain pathway which is linked to genes which are at this side. So I have this kind of curve over here, and this brought me to this result where I have a statistically significant pathway. That's why it got a tag. Statistically significant pathway for downregulated pathway because of my expression profiles of my genes being related to this pathway. And then I can go and browse it and see it. There I have different options. So I can, for example, show only genes which are differentially expressed, and I can see those different expression values in my results down here. And I can also paint the expression data directly on the map. So I do not only have to paint whether if it's there or not, but I can see how the experimental design looked for each enzyme linked to a certain gene or transcript. Okay. And then within this view, I have a lot of information about the statistical significance, about the linked enzymes, and how many sequences are linked to each enzyme present. And then I can actually search for enzymes or genes or certain pathways and switch the view mode as well. So, and this brought me actually to the end of the introduction of all those features and functions available in OmicsBox, and particularly for the functional annotation and the pathway analysis. And I think we have still a good some 10 minutes left here for our demo, which is great. So let me jump into OmicsBox here. And I hope you can all see my screen now well. And big enough to read all those different things here. And yeah, so this is OmicsBox now. And let me try to show quickly a few things. So let's open up, for example, our mice data. So we have raw counts here. So So we have the raw counts here, and I can see that's just a subset. Let me see if I have the full set. See, I opened one which was not the one I wanted to show. So now here I have all, you remember we had our 12 samples here. So I have my 12 samples and I can sort them by counts. So that's actually 40,000 rows. OmicsBox pretty performant here on sorting and filtering. So you can select greater than, not equal, less than for different conditions here to subset your data. And here are actually my gene names. So remember, the mice data was actually a model organism study. So here I have my official gene names. So when I'm looking for my functional notation, I also have to have those gene names. From my side panel, oops. From my side panel, I can see my actions I can do. So I can rename features. I can select sequences. For example, a group of genes I found. And I can directly hop into the difference expression analysis. So, for example, here I would have the option running DSEQ, DSEQ2 actually. So I have my experimental design, which is just a text file with a couple of tab separated columns here. So I have my stages, virgin, pregnant, lactate, and luminal and basal. And then that's basically, as you can see here, it's just a text file, which is called design.txt. And then I just click next, and then I have my genes filtering, which is counts per million, total counts, samples reaching the filter here. You could set this to 3. And then I click next, and then I have simple, multifactorial, or interaction design. So I have a blocking factor which I can set. I have okay, I don't really want to spend too much about this because you know about how to do differential gene expression analysis. Just wanted to show you that these are downstream analysis options here. So I have HR and pairwise. And once you have that running, you can go and open up, for example, this one here, which where I was comparing lactate versus pregnant samples of only one tissue, the luminal tissue, okay? And then I get this table, and I can directly filter my accounts for up or down regulated ones. And then I can see up and down regulated text. And if I filter for the FTR, I can see they're getting mixed up, and I have those list of genes. Okay, so now I have the summary report. I can rename features. I can set my up and down regulated text to, let's say, 3 for up and 3 for down. And then my numbers over here change if I recreate this report. So now I have 180 up and 190 down regulated genes. And here I can see my experimental design and everything which I performed here. And from there, I have my charts. So let's say, for example, an MDA plot. Let me show you on the raw counts first. So I have a PCA plot here, and I have my experimental design, and I can say cell and stage and a 3D plot. And then I can very nicely plot my data, check for batch effects, and see that my couples here of lactating, pregnant, or virgin group very nicely together. And then I could maybe change colors or change symbol sizes, those kind of things, right? Same for distribution of counts, for example. This is like kind of immediate stuff you can do. It's the same, for example, for the difference expression results here. Okay, so now having that, let's try to do the functional analysis here. Okay, so obviously, as I said, for the functional analysis, we need functional information. So what I did is I went into the functional analysis and I did load data from Biomat. I opened up Biomat and I put in vertebrate, genes, and I went to human genes, which one? This one. And then I decided to take just the standard gene names here, stable gene names. And these are the ones which I already have also here in my feature list, which is important. Those ones have to match. And then I just take the whole genome and hit load. And then this imports me my annotations. And then I just get them like this. In this annotation project, I already showed you, so now you can see this in action. I have my project here, and then for each sequence, I can show you the gene ontology terms, which are listed here, okay? And as you can see, some have just one geoterm, and if I scroll down, some have more, some have 4, some have 6, different categories here. And this is what you get when you import annotations, which is just gene ontology terms. So in my context menu, remember, so I don't have blast results, I don't have mapping. I did an InterPro scan on it because I actually have my sequences here imported as well. So I can check on those as well. So now I have my annotations. I just imported them. And I have my difference expression analysis. So now what I can do is I can go from the side panel directly into my enrichment analysis, for example, and I can pick a Fisher's exact test or gene set enrichment analysis, and I hit run, select my ranked list, take this annotations here, and then run it. But I could also go directly from my paired end result, and I can go into the actions, go gene set enrichment analysis, click here, and then I select my biomat ensemble annotated genes, the blue ones over here, and then I hit next, 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 and run. And then I get my gene set original analysis started, sent into the cloud, get all my permutations tests done. I see my progress over here, my details over here, my application messages over here, and then everything gets calculated. And within a couple of minutes only, let me see, I will get, using the functional information and the difference expression analysis, I get, for example, my lactate versus pregnant luminal GSEA. Okay, so here you can see I get again a table. I get gene ontology terms. I get tags for bottom and top. And if I right click and say show details, I can see that this is a down-regulated term, heavily present among down-regulated genes over here. So I get this enrichment plot over there, statistically significant for this for this molecular function, which is actually structural constitute of ribosome. I can look at this. This is a very high-level term, so maybe not the most relevant here, but I can get a lot. I get 690, and I can perform an enriched graph here, but I also can reduce it to the most specific ones. Remember, I can remove those intermediate ones if there's a statistically significant one at the end of my graph, so the most specific one. So if I reduce those by using a FDR of 0.05, I can go and run this, and I get again another table of the more specific ones. Maybe I had been a little more too restrictive here, but I get this kind of result then there. Charts are available, as I showed, like bubble plots and word clouds and those kind of things. Not sure why this doesn't work. And the bar chart as well here. You see, like this. Okay, perfect. So this is the standard stuff. Now let's go into the pathway analysis. So the combined pathway analysis brings in together everything we have already seen until now. Okay, so here we can see how we can take our sequences, which are annotated or not, and combine them with our difference expression data. We thought it over here, our pairwise expression data. And then we pick an enrichment analysis, which could be gene set. Oh, cool. See, the GSEA results just came back over here. Let's cancel that. Let's see those ones. Those ones got just created. Let me see if I can get a bubble plot now. And let me see if I can get a word cloud. Yes, that's all working nice. Okay, so you can already look at functions being up and down regulated among your difference expression set. So back into the pathway analysis here. So we pick our annotated genes, and we pick our difference expression data. Remember, very important, The names have to match, okay? Names have to match, but you can use batch rename features to get that done. You can also match them by sequence similarity. That's more advanced, maybe for another webinar, but that's something you can do as well. And then we have different pathways. We have the reactome pathway, we have the Gramini for plants, and we have KEGG, okay? Let's stick to KEGG for a second. So to link your sequence data to your pathways, that's a tricky step, and OmicsBox offers different options. So you can basically just take your enzyme codes from your annotations and search them in the KEGG pathway database, and then you get these links. If you don't have enough enzyme codes, or if you want to add additional information, you can search your sequence data in EggNog, identify KEGG pathways via EggNog directly, and then link them as well. And there are filtering options for specific pathways you might not want to include. And for the EggNog option, you can choose whether you have proteins, CDS, or genes. Okay, this is a little bit different than how the search is performed. You can read it up in the help section for each of those parameters. Once you click next, then you have this cloud sync data handling option, which is new in our latest version, where you can store a local input in the cloud. You can save results in the cloud as well, and then you get email notifications once a job is done, and you hit run. Okay, so this. Oh, sorry, I didn't show the reactome, which is also interesting. Let me open it up again. So for the reactome one, it's slightly different. The reactome pathway database. Here we are diamond blasting our sequences to the reactome database. So all the sequences which are present in the reactome database, we are searching them against this. So annotating it by that, plus all the gene ontology terms we already have in our annotation set. If you want to stick to the official ones, you just go like this, and that's super fast. If you want to add in additional information, just similar to the EggNog, here the blast option, you add this, and you can link additional information. You hit next, 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 and run, and then you get your pathway results. So let us look at the pathway analysis for luminal pregnant and of lactate tissue, luminal tissue, pregnant and lactate mice. And then we get this table here. So you see 424 pathways could be found. And then epidermal growth factor. Let's see if there are anything related. Let us use the search function here. I typed it wrong. Okay, so now I filtered this for every pathway with epidermal surge growth factor, and I saw that I get a couple of them which are completely, which are statistically significant, and where genes are downregulated. So, for example, let's look at this MAPK signaling pathway, which was also mentioned in the paper. So let's go into the context menu and look at the pathway diagram. Once you found something interesting, you can go to retrieve selection map data of a certain pathway, and you can find linked genes, difference expression of linked genes, the gene ontology germs or enzymes or EggNog orthologs which are related to this selection, and so on and so forth. Okay, so let's go into this pathway. Let's directly make it big. And I'm kind of running a little bit out of time because I'm already hitting my 1 hour limit. So just bear with me for a couple of minutes and then I will wrap it up, okay? So this is my MAPK signaling pathway. And as you see here, for the different enzymes, a lot of genes are involved in my data set. I already can see, for example, here at the bottom, that I have my luminal ones in the lactate stage being downregulated. That's the responsible ones for having this result here. Let me see if I can remove this again. And now let's try to see map the expression data. Okay, wow, that's kind of overwhelming. A lot of information here. Let's see only the differentially expressed ones. Okay, that looks much better. So now I can look through my list and I can see a lot of candidates here, which share kind of a very similar pattern. And now it's kind of up to you again to search for certain things. You can see different options here. You can search for them, and then you can identify things on your map. Uh, and then you can dig deeper into this data, see which pathways we are having, which genes are related here, filter, change the visualization for expression or for reactions. So this kind of stuff all readily available. Maybe just to see another one, so you can see different visualizations of those pathways. Here again, let's search for the epidermal growth factor. Let me see if there's something. I could just see this. Let me see it on the map. Okay, here we are. Okay, so here I have this one gene being completely upregulated. Uh, chemotactic factors here. So, and then I get kind of lost in the biological interpretation, but we can see that it's directly links back into the MAPK signaling pathway here. So maybe that's something which gives you hints. And this could be actually done for any of those pathways here. You should focus on the ones which have a lot of differentially expressed ones, but you can also, and remember the statistical significance here is always only a cutoff. So you can widen that, you can change that, you can redo the difference expression, redo the enrichment analysis, you can redo the difference expression and exchange data, so you don't have to run the pathway analysis all over again. So you can just rerun the difference expression and then put it in here, or rerun the enrichment analysis and put it in here. And obviously, you can also export your data, export pathway diagrams, or export a dynamic data as well, where you can choose specific columns. And remember, columns in OmicsBox are configurable, and you can visualize species codes or classes or a lot of other things in OmicsBox here as well. So I didn't really show the nematode dataset a lot. It's in terms of the pathway analysis, it's kind of similar. Maybe the big difference is that you had to run like through the whole annotation process of your novel transcriptome. And so you were not able to like just go into the load data from Biomat to get your annotations, but you had to generate them from scratch, and you had to make sure that your IDs used in your pairwise difference expression analysis do actually map the annotations here. And then something which I always highly recommend is to use the Venn diagram. Let me see if that's working out of the box. So when you're working with lists from different type of data sets, so for example, a data set for annotations and a data set for expression data, so those IDs not always match. Or not always cover the full data set. So for example, you might have much more genes here, for example, than in your annotation, or the other way around. And then you want to see for how many genes do I actually have functional information. For the human ones, that's basically you have what you have. But for the transcriptome, where you generate the annotations from scratch, you can really then make wise decisions and say, okay, with the annotation filters I used, I only got 50% of my genes being annotated. So let's at least make sure the most up or down regulated ones are functionally annotated, so that I can get a proper downstream enrichment analysis for those kind of things. Okay. So wow. Okay. So with that, I'm getting back into my presentation, and I'll finish my demo here. I have just a few closing remarks for the webinar of today. So take-home messages. So you get this end-to-end analysis with OmicsBox, so you can bring your raw data up to functional insights, especially with the combined pathway analysis feature we showed today. It integrates and maintains a lot of different tools and databases. We take care of that, so you don't have to worry about it. You just put your data in, and run through your pipeline. So the hard bioinformatics background work has been done for you here, which also includes maintenance and it includes support. So you're always welcome to write us and ask questions. As you saw, we can do that for model and non-model organisms. So mice, human works fine, but also nematodes or plants or fish or whatever you might have. OmicsBox is great for the generation of and combination of different functional annotations and able to actually map it back to your expression data and then put it onto pathways all together in this combined approach, which is kind of cool, I think. And then OmicsBox allows you to easily combine everything because you have this downstream side panel analysis option. So you just click from a pairwise into a Fisher or from an annotation into a pathway analysis, those kind of things, no? And then I hope I convinced you by showing all the rich visualizations. So this is something we definitely offer and have. Okay. So a quick teaser for our upcoming release. It's just a couple of days left until we will be able to show that to the world. So we present Cloud Sync and the OmicsBox web as a first. And a lot of different bioinformatics analysis options also for long-read transcriptomics again. We have new stuff for single cell. We have our core expression network data analysis, pretty new, fancy networks, linked to gene ontology terms for core expression, and many other general usability advantages we had been improving over the time here. So just to introduce CloudSync briefly, it allows you to work better with the cloud, save data into the cloud if you want, and run data offline. So you can send things away, wait for results, and then shut down OmicsBox and get back a couple of days later and then recover results from there. So the disconnect and the sharing data options are the most or the best or the coolest things here, apart from the new table where you can monitor much better what you send away into the cloud for computation. So thank you very much for having stayed until the end. I know.
Thank you for watching. Learn more about OmicsBox with our other videos.